All right. So those are the four mechanisms. There are other mechanisms that you can study in biology. A big one is sexual selection also, but you don't really get into that in this class. You will have to learn it in evolution class. But remember, our definition of evolution is a change in a population's allele frequency over time. That's it. And so if you acquire a mutation, your population has evolved because that's a new allele. So it changes the frequency of the other ones. Okay, gene flow is individuals, genes really, moving into or out of a population. Okay. Genetic drift is random selection of variation, and natural selection is non-random selection. So let's get into natural selection here. It can go a few ways. The one we typically think of first up is directional. Can somebody give me an example? Directional selection.
Like the beaks and the finches from the Galapagos. Like when only the large seeds were available, the beaks had to be larger to crush them.
Perfect. So we've got a selective pressure, which is the seed size. Okay, and the birds with smaller beaks got less food, so they made less babies, fewer babies. Birds with larger beaks, that mutation got to make more babies, and so it pushed it towards that larger beak trait. And so that's directional, is pushing towards a single version of a trait. Okay. Disruptive, who wants to take that one?
Would this be like maybe there's like 3 colors of flowers in a population, but then one of the colors started to be less prevalent than the other 2?
Yeah, perfect. So let's say we got white and red flowers and also pink. Okay. Assuming that this is like a nice independent trait and everything, all that good stuff. If the bees are more attracted to the white and the red and not the pink, then that's the environment selecting for white and red. So it's pushing towards the extremes of a given trait. Okay. And then stabilizing. Can somebody help me with that one?
That would be like the antlers on moose or deer, because if it's too big, or I don't know if this is a great example, but if it's too big, then they get caught up in stuff and then they can die because of that. And if it's small, then they don't get mates.
I like that example. I hadn't thought of that one. That's perfect. Yeah, so it's pushing towards like kind of the middle ground for a trait. And then I think the toughest one is balancing selection.
Is that also known as frequency-dependent selection?
Frequency-dependent selection is one type of balancing selection. It's like a special case.
Is that when the environment is selecting for exactly what's present?
Can you elaborate? Maybe I misunderstand.
Just so that there's no pressure to evolve really one way or the other. It's what's present is what's ideal.
I see where you're coming at, and maybe I'm gonna give you a maybe on that one. We'll look at a couple examples and see if you.
With one of the questions on the quiz with what was it, malaria and sickle cell, would that kind of be like that? Like
yeah. So
because if there's more malaria, then it's better to have more like sickle cell, and if that's the thing that's killing them. But if not, then it's better to have just regular. But
yeah. So let's take a look. We'll start with that example right there, because it's not good to have sickle cell anemia. But remember that trait, you can be heterozygous for it, and it's still kind of bad for you unless malaria is present. So we got a couple different ways of looking at balancing selection. It's the heterozygote advantage that we're talking about right now, and then frequency dependent selection. So heterozygote advantage, somebody unmuted and crunching paper, just FYI. Heterozygote advantage is one way to kind of balance, to keep sickle cell anemia around. So maybe that's kind of what you were saying about how like it's selecting for what's there. Balancing selection is keeping traits there, keeping both versions of the allele. Okay. So what's going on is in most parts of the world, it's really good to have 2 copies of normal sickle cell allele. In other words, you don't have sickle cell from either parent. Where there is malaria, it's still really bad to have 2 copies of the bad variant, sickle cell anemia. Okay, you don't live very long. You have a lot of health problems. But if malaria is present, like in sub-Saharan Africa, then it behooves you to get a good copy and a bad copy, because you're kind of getting the best of both worlds there. You're getting immunity to malaria, and you're not as sick as if you had 2 bad traits. And so this is a heterozygote advantage. Those with a good copy and a bad copy have the best fitness in this environment. Okay, so they're going to be passing on that bad allele to their offspring over and over, and so it's keeping it there. Now, the other way of looking at balancing selection, another good example, is frequency dependent selection. So your textbook gave this example of the fish, this parasitic fish. All right, and they're kind of weirdos. They're not born with a mouth right up front. They're born with their mouth on the left side or the right side. Okay, so there's 2 alleles. There's 2 variants in this population of fish. Which is it better to be, a left side mouth or a right side mouth?
Doesn't really matter.
It kind of does.
Probably
So
depends
you're making,
on which side the prey has, like which side the prey has armor each year.
oh, okay. It's got it on both sides.
Okay.
So you're this little fish attached to the side here. So you make a living by latching onto the side of a bigger fish and kind of drilling into its flesh and slowly sucking out blood and guts. All right. This makes sense to have your mouth on the side if that's how you make a living, because it's more aquadynamic, right? That fish can keep swimming along and you're just stuck to it. How many fish, these parasitic fish, can fit on the side of one of these bigger fish?
Probably only a few.
Yeah, usually 1, maybe a few. So just a few. All right, so let's say you're born with a left side mouth. What side of the fish can you attach to? It's right side. All right, so at the top in this little chart, that's a left mouth fish. Okay, it's taking up valuable retail space right there. Okay, it's found its real estate, and not many more can attach to that fish. All right, so now which is it better to be, right or left?
A mixture
Now
of
it's better
both.
to have the right.
It's better to be the right if everyone else around you is a lefty. Okay, because that means you've got more food choices. You've got lots of open real estate on all these fish that you're parasitizing. Okay, so 2 years ago, this big flush of babies came out, and they're all left mouths, and they go out and they find their fish, and now you're hatched. You're a right side mouth. Things are great for you. Things are less good for any babies that are hatching that have a left side mouth, because there's not much room for them. There's not as many resources. Okay. And so suddenly we get natural selection pushing all the babies to be right-mouthed, because that's where the food is. Then what happens?
Then it flips back to everyone wanting to be a left mouth.
Yeah, because now natural selection's like, okay, all of you, right mouth is better. So left mouth ones are dying. You right mouths have a field day. And then suddenly everybody's a right mouth and it goes back. It's suddenly better to be a left mouth at that point. And so it's dependent on the frequency of others that share your same phenotype. And so that's what this chart is showing, this staggered up and down. And we can observe this in nature. The proportion of left mouth to right mouth goes up and down year after year. Okay, so like every 2 years we get a reversal.
Would the evolution of metamorphosis in insects be another example of this? In the sense that, like, the benefit of one species literally looking different and taking a different ecological niche when it's the young of the species versus the old of the species allows for way more of that species to exist.
Oh, maybe. I think it's a bit different because it's not going to be going back and forth there. You're talking about a way to kind of split your resources between your young and your old, which is a different thing. That's competitive avoidance. And we won't get to that for about a month, but you're on the right track for sure. Now, is anyone familiar with cicadas? We don't really have them in Utah, but
Oh yeah.
Unfortunately, yes.
okay, yeah, they're insane. All right, the cicada life cycle, they can spend 7, 9, 13, or 17 years underground feeding on sap from roots, and then they emerge all at once over a space of like 3 days and start screaming in the forest, like ear splitting. It can be 120 decibels. Okay, but they all emerge at once, and they've really timed their emergence patterns to kind of avoid each other, and so that's frequency dependent selection also. So that would be a kind of an insect example. If all the cicadas, the different broods, were emerging at the same time, there'd be not as much to go around. Okay, not as much tree space, and they'd be running into the wrong mates from the wrong species and all that. And so what we see now with these crazy staggered cicada emergence patterns is the result of long time of balancing selection and frequency dependence. Right. Another example is the flu virus seen here. You may have heard flu referred to by a couple letters like H1N1, H2N1, right? What's that talking about?
Isn't that the proteins that are the way they identify it essentially?
Yeah, it's the like spike proteins on the
Yeah,
outside, for
that
sure.
thing.
Okay, well, if you get the flu, your body develops antibodies to it, hopefully, if you don't die, and now you're immune to that strain. Okay, and so we have a strain of flu that will go through the United States 1 year. We don't get the same flu next year. It's not because it's not around in nature, it's still out there really, but everybody's essentially immune. We've kind of gotten a herd immunity to that particular strain. Okay, so the next year, a new strain is going to come through. And so this is frequency dependence as well. The surface proteins on the flu virus are changing, and 1 year it's good to be H1N1, the next year it's good to be H2N1 or whatnot. Okay.
So is that what's happening with the coronavirus, with the variants?
Yeah, the variant there, it's this spike protein on the capsid, and so it's pretty similar in terms of how it behaves to the flu. It's just a lot worse. You know, it's really new, and so we don't have any defenses for it. Okay. But yeah, that we have noticed that new like UK variant that has had, I forget the number, but just a few amino acids have changed in that spike protein, and that's made it way more transmissible and contagious. Okay. And so now, yeah, you can see the coronavirus, that's going to be the strain that most people have pretty soon, not the old one from 2 months ago. All right, any questions about any of these types of natural selection? All good? All right, well, let's see if you can identify them. I'm going to tell you a story about some mice that live on an island. This is the initial population of mice here, and they're living on this island. We've got roughly a third for black, white, and brown mice. All right, so that's our allele frequency to start out with. However, their island, they share it with this really evil, angry wet cat, and it likes to kill them. And so some of these mice decide to set sail for a better land. Okay, so this is our population that gets in a boat and sets away. What happened?
Isn't that gene flow? Or
Yeah, it's a special case that you saw in your chapter.
Isn't it called like the founder effect?
The founder effect, yeah. And so we're taking, you can argue about whether it's random or not, but let's, for this sake, we're saying that there's a random selection of mice. It didn't matter whether they were brown, white, or black for deciding to set sail. It's just these are the ones that did. Okay. So they set sail.
Is it
The allele frequency has changed.
is it still founder effect if it's one of each species? Because I always thought it was just like one species that left to become the founder. Or
Well,
is it
this
just any
is
of-
one species of mouse. It's just 3 different colors. So
Oh, that's right. Sorry.
Yeah. So evolution, we're always working with populations, not even at a species level. It's populations are the things that evolve. All right, so this is the founder effect, and we've gone from roughly a third of each to now it's half white, a quarter brown, a quarter black. All right, so this population has evolved through a special case of genetic drift called the founder effect. All right, so they show up at the island, a new home for them. It's full of white and black rocks, but there's evil birds there that are killing the mice. All right, so what do you think is going to happen?
disruptive selection.
Disruptive. Why?
Because it's white and black rocks, but if you're in between in a brown color, then you can't blend into either.
Yeah, the white and the black mice have places to hide. The brown ones are screwed. All right, so after the 3rd generation, we're down to 4% brown, 60% white, 40% black, roughly. Okay, so there's our proportions now. Okay, so that's natural selection, and it's disruptive. Now, sadly, it turns out that the island was also a volcano, and it erupted, and it killed most of these mice. All right, what kind of evolution is this?
Genetic
That's,
drift.
yeah, genetic drift.
Genetic
Sort of.
drift. All right, your phenotype does not influence whether lava kills you. You're definitely going to die. Okay, so we accidentally lost the brown allele. It's gone from this population. That allele is said to have gone extinct. Okay, at least in this population. So it's really sad. There's no brown mice left, and our allele frequency has changed yet again because of genetic drift. Okay. Now some new immigrants arrive on the island for black mice. What kind of evolution is this?
It's gene flow.
Gene flow, yep. So we've got some gene flow, and now we're 50-50 black and white. Okay. And then, sadly, the cat swam out to the island and killed them all. That's the end. So looking at that, we can actually kind of track alleles over time in a population. Okay. When we do this, we've noticed that genetic drift is much stronger in smaller populations. Let me go to this simulation here. All right, so let's start out 50-50. Can you see this? The website?
Yeah.
Okay.
Yes.
So we're starting out 50-50 with 2 alleles. And we got a population size of 100. Let's evolve. So this just is genetic drift over time. You can see that it's moving towards one allele. You'd be tempted to say it's directional, but this is random. Oh. What if we made this population size 10 individuals? See how the fluctuations are bigger? And we lost an allele. Okay, what happened? Why is it stronger in a smaller population? Oh, I just interrupted somebody.
Because it was a smaller population, the effects were much greater.
Why?
There's less population to balance it out.
Okay, so if we go back to like our little brown and green beetle example with the guy stepping on the green ones, there were only 7 beetles in that population, 7 individuals. And so if a footprint covers 3 of them, what are the chances that you're going to wipe out a big proportion of the green ones? Pretty high. Okay. It's just random chance. You may have just stepped on brown ones, but you're still changing a big proportion of the overall population. Now, if we set this to a population of 10,000, you can see that if you're just randomly killing off some every generation, that's not going to have much of an effect overall on the allele frequency in that population. You can still see that it's changing, but it's hovering around 50-50 because that's where we started. Okay. So in a big population, get back to my screen here. In a big population, allele frequencies look more like this on the bottom image. And in a small population, they tend to do this because of genetic drift. Okay, so if an allele reaches 100% in a population, that means that everyone there has that exact allele, and so there's no more variation, at least at that genetic locus. So we call that fixation. So one allele goes to fixation in a population, and then you see it stays there for presumably ever, right? Because everyone in that population has that particular allele. There's no way you can pass on a different one to your offspring, and so it's going to be fixed.
Well, what about mutation?
Yeah. So mutation is the only way to get out of that fixation. Okay. These particular simulations we looked at didn't incorporate mutation into them. Right. Okay. We good on genetic drift? Okay.
Yes.
All right. Until I hear otherwise, I'm going to press on and tell you a story about evolution. All right. This is a human and a chimpanzee. What's different?
Chromosome 2 is in 2A and 2B.
Yeah. Okay, well, chimpanzees, orangutans, all these, the great apes, They all have 24 chromosomes. Humans have 23. And it's long been a mystery, right? So, okay, if humans did evolve from a common ancestor with chimpanzees, shouldn't they also have 24? It would make sense that they would have 24 pairs, because all the other great apes do. So what happened?
Did the 2A and 2B chromosomes join up?
That's a hypothesis. Okay, so we've made our observation here, nature science-wise. There's a fact. Chimpanzees have 24. They're our closest living relative right now. We're sister to them on any tree of great apes. The other observation is that humans have 23. Then a hypothesis might come around, okay? And the hypothesis is a potential explanation. And so you've said that 2A and 2B have fused up in humans. Okay, what's a hypothesis good for?
Proving things wrong?
Yeah, proving things wrong, exactly. So technically, what we want to do is try to prove the hypothesis wrong. All right. So we've got our potential explanation. It needs to be testable and falsifiable. If there's no way to prove it wrong, it's not a valid hypothesis. It's a waste of your time. It's not part of science. We don't deal with that. Okay. There's any number of infinite explanations that could have gone into this. Most of them aren't testable. This one should be. How would you test this?
Could you sequence the 2A and 2B chromosome and then sequence the second chromosome in the human genome and see if they line up?
Okay. They should be able to line up. There's going to be differences. I mean, if it was exactly the same 2A and 2B that just got fused together and now we're humans but the sequence is exactly the same, we would still be chimpanzees, right? We do have some differences in our genome from chimpanzees, so you're on the right track. So let's look at chromosomes. You've seen these before. Here's one under an electron microscope down at the bottom. And chromosomes in eukaryotes have this very distinctive structure. We've got telomeres on the ends. We've got a centromere somewhere towards the middle. Okay. Anyone know what the centromere is for? Well, how do you do meiosis, sexual recombination? You've got to like join them up, and so it's this kind of like, think of it
binder.
Like, yeah, like Velcro. So it's, if you get rid of the centromere, sexual reproduction doesn't work. Okay, so that's conserved. We call it conserved. All these eukaryotes that do sexual reproduction, they've got a centromere. Okay. And then we've got telomeres. What are they for?
They fill in the extra when you're sequencing, or when you're, is it in transcription or translation, but it's filling in the gap in one of them. I forgot where.
So it's actually during when DNA polymerase joins there and it's copying. So like the mitosis part, when you're copying a chromosome for cell division, the way that eukaryotes are set up, because we're not as good as bacteria at this, is that we have these long end pieces and they slowly get chopped off. Every time there's a copy, you lose some of them. Okay. So those are all the same in every animal, every plant, all that. And we know what telomeres look like. It's this sequence, this long repeat sequence. Okay. All right. As you get older, your telomeres shorten. There's hypotheses out there that that's part of senescence and aging, is the shortened telomeres. And this is the centromere. That's the code for it right there. All right, so we know what that looks like. We know what the telomere looks like. And so if our hypothesis is the missing human chromosome is actually stuck inside another chromosome, that should be testable and it should be falsifiable. And this figure right here is basically an image of the hypothesis. All right, so it's making a prediction. What specifically is the 2A and 2B fused together? What's that prediction that we can test?
We should find a telomere in the center of one of them.
Yes. And so we should look at chromosome 2 in humans, and if we sequence it, we should find an extra long double length telomere right in the middle, where it has no business being otherwise. Okay, so that's what we did, right? We've got chromosome 1 here. This is human, chimpanzee, gorilla, orangutan. Okay. Chromosome 2, human, chimpanzee, gorilla, orangutan. We just call that 2A and 2B for those. All right. What we found is slightly confusing, but let's take a look at it. We've got our centromere. We've got a degraded centromere that's not really used anymore. And then did we find our telomere there? In the chimp, we go telomere, centromere right there. We've kept the same centromere as the chimp 2B chromosome. The chimp 2A chromosome, that one has degraded. It's acquired a bunch of mutations over the last, I guess, 4 million years since this switch happened. Okay, 4 to 6. And then there's our fusion point, and we found our telomere. And so what we actually found was what the hypothesis predicted. Okay, so it's exactly what it predicts. What about the actual genes on there? We should have the same, we should like be able to take chimp 2A, look at these genes, and they should match up, like you said with the alignment. Okay. And they do. It's the same genes, just in reverse order as you're reading down. Okay, so this lines up perfectly, and it also generates new predictions. Okay, so we put our hypothesis through a test at this point. How did it do?
It did pretty well.
How well? Good enough that you're gonna accept it?
Well, we were right, so yeah.
Okay. Well, how about this hypothesis? If I flip a coin, it's always going to be heads that comes up. And I flip it once, and it's heads, and I say, all right, hypothesis tested.
What I mean is, what are the odds that the one human we picked happens to have a fused chromosome? Doesn't that seem really unlikely?
Yeah, and we've done it with like 700 humans at this point. Tested this very thing. So that would be very, very unlikely. All right, so the point I'm trying to make here is that you test a hypothesis. Sometimes the test is so strong and unlikely that if it comes up right, the hypothesis seems pretty believable at that point. This is one of those cases. The coin flip thing, that's not very strong evidence. All right, I would say test that hypothesis again. Do it 100 times. Do it 1,000,000 times. If it comes up heads every time, then I'll start to believe that hypothesis is reality. Okay. So we've got our hypotheses. We try to disprove them. We looked in there. If we hadn't found this, we would have tossed out the hypothesis. But we did find it, and it's so unlikely to be what we would have seen if this hypothesis wasn't reflective of reality that we accept this hypothesis tentatively. That's key. Tentative acceptance of this hypothesis. Okay. We could get more information that makes us change our minds. I wouldn't bet against it, or bet on that, rather. This is really strong evidence. All right. Now, final thing today, before I take your questions, is this concept of natural selection having foresight, and the fact that it's limited by historical constraints. So if I say natural selection is limited by historical constraints, which we've kind of seen a nice example of that, what am I talking about?
It can only build on pre-existing structures, I guess.
Yeah, well, to a certain extent. It can build on pre-existing structures, but the environment can only select from things that are already there, so yes. But it can also be influenced by mutation. So we're going to take a look at a giraffe dissection. All right. Hopefully this video doesn't give us all seizures or anything, but trigger warning, you're about to see a giraffe being cut open. Whoa. It's kind of trippy.
We forget that sometimes they're not perfect and there are imperfections. And the imperfections are very revealing because they're exactly the kind of imperfections you'd expect from the accidents of history if there were no designer. There's a nerve called the recurrent laryngeal, which runs from the brain, and its end organ is the larynx. And you think it would just go straight there. But in a human, what it does is goes down into the chest, loops around one of the main arteries in the chest, and then goes straight back up again. Obviously a ridiculous detour. No engineer would ever make a mistake like that.
In our bodies, the laryngeal nerve takes a circuitous route. What happens in an animal with a neck as long as this? It's one of the great evolutionary enigmas that Richard Dawkins is keen to resolve in The Flesh. Interestingly, it's never been dissected out, or only once before has it ever been dissected out in its full length, and that was in 1838.
Don't cut it at this crucial moment.
Too good anonymous for that. You have to trust me now. What I'm actually holding here is the beginning of that nerve. It actually starts out not as a separate nerve, but as a branch coming off of a bigger nerve called the vagus nerve. And this is going to keep running all the way down the body. So you'll see it again over here, all the way down the neck on both sides. So you can see it again right there.
Yeah.
And this is going to wrap around the great vessels coming out of the heart. So here it is wrapping around that, and then it continues right there. So here's the vagus going down, and here's the vagus continuing. And right over here, there's a branch right there. So it's looping and it's coming back, doing a U-turn all the way down here. So it's traveled that entire distance to make a U-turn to go all the way back up again. And so now we can follow it going back up again. So we follow this branch, and if we look and see it again over here, there it is, like that. And here you see it going up. This is the voice box, the larynx, and so you can see it going right into the back of the larynx here. And as it innervates this area, it controls the muscles that then control making sounds, but also coordinating breathing and swallowing in this area. So this is a very important nerve. Interestingly, where it ends is pretty close to where it started. It started here, coming out of the brain. It really only needed to go about 2 inches. But it went all the way down and came all the way back.
It's a beautiful example of historical legacy as opposed to design.
Okay. Historical legacy for the recurrent laryngeal nerve. So if you caught that from our trippy seizure-inducing video, it's a nerve that runs from the brain to your voice box. And it controls speech, swallowing, coordinating breathing, very important thing. Why is it that it starts here, goes down, loops around a ventricle of our heart, and then back up?
Other bones of ship around it, and I think a lot to do with how the animal moves and stands upright at different angles?
No, it's actually not a good thing that it does go around there. It being such an important nerve, the longer you are, the more chances there are to kind of mess up. And we actually see this in human embryos. If it doesn't form correctly because it's a long journey, then typically those babies are miscarriages. They don't make.
I mean, like the giraffe is evolving from a mammal that had a different skeletal structure, and then as it's turning that skeletal structure to accommodate a different posture, it gets potentially twisted up in that area.
Okay.
It's twisted in humans too, though.
It's twisted in humans and mice and dogs.
Okay.
And
could it be like
snakes, lizards?
Could it be caused by evolution over time? I mean, like the nerve, as the body evolves, gets just like accidentally caught on the wrong side of the heart or something? Or is just like accidentally put on the wrong side of the anatomy by some other structure developing?
Huh. Okay, so then when you elongate over time, it still kind of has to go that route.
Right.
Okay. Fish have this nerve also. It's homologous in fish and all vertebrates.
Is it because of the spine? Is it just to get the nerve out of the way of the spine to make sure it doesn't get crushed by anything?
Oh, no. It's a good guess, though. Really, if you were to like redesign vertebrates from the ground up, you would put this nerve going a half inch to where it belongs. A lot of our other nerves take the shortest possible path. This is like that weird one that people couldn't explain.
From my understanding of this, from a very old or early common ancestor, this was the shortest path. And then as they kind of evolved, it's okay, let's just make this nerve, a little bit longer. And then everything else changed where it was more efficient to just, make it a little longer than to like redesign the entire thing.
Boom. Perfect answer. Okay, so in fish, it still is the shortest path to where it needs to be. But fish don't have a neck. Okay, so it does loop around the heart. When tetrapods, came onto land and all that good stuff, and necks became important, it was a lot easier for evolution to just elongate this nerve rather than completely reroute it. Okay, think of all the mutations that would have to happen to, like, let's say that you have a kid and your kid is born with a good laryngeal nerve that only travels an inch. How many mutations would have had to have happened to get that? Well, you'd have to have the mutations to delete the entire laryngeal nerve perfectly without messing anything else up, and then all the mutations to create a new one. So millions of mutations would have to happen perfectly, basically overnight. The odds of that are just staggeringly large. Not going to happen. But how many mutations does it take to elongate a nerve that's already there? One. Okay, and so it's much easier to work with the nerve being in that stupid path now that you've got a neck. Okay, so this is historical legacy of evolution. Hopefully that makes sense. It's a good illustration of how evolution doesn't have foresight. It didn't plan for necks when the laryngeal nerve first pathed its way around the heart. Okay, it wasn't thinking ahead. And so we're left with the nerve as it was originally routed, because that's how all vertebrates develop. So in developmental biology, you can see this nerve forming, and it takes that little path, and then the neck grows and the body elongates So that was the last thing I wanted to cover today. Friday will be wrap up on this topic and time for your questions. So please bring your questions about the material for Friday. Oh, look. This is still evolving. Forgot to stop it. Okay. So, anything else?
How do cicadas all come out of the ground at the same time? Is there some kind of like colony signaling going on, or?
It has to do with like an internal clock, actually, in each of the broods. I can't give you a better answer than that because I don't think we have one, and I just read up on cicadas for
So like
fungal research thing, and it's still kind of like a mystery.
So more like a certain kind of species will go every 7 years and another kind will go every 14, not like this year we're going 14 boys.
Yeah, yeah, they're different. They're different species, for sure.
Okay.
Yeah.
Cool.
And even there are some cicada species that have different broods, and so like different populations of them. One of them's on like a 9-year plan. The other one's on a 13-year plan, even though they're the same species.
Cool. It seems like something like I can't remember the word, but viruses or some bacteria where when they get to a certain level, they just like, yeah, quorum sensing. Thank you.
Yeah.
That would have been
no.
pretty darn
I think we've disproved that hypothesis for cicadas, and we don't have
makes sense
a good mechanistic explanation as far as I know.
Cool. One can dream.
Someday. I mean, you could go to grad school and get a PhD in cicada timing. That could be your life. They'll pay you to do that. If you really care.
As much as I like circadian rhythm, I like the idea that every cicada just has Wi-Fi.
There appears to be no communication between them because it's like entire forests just all at once emerge. It's quite a sight to behold. All right. Well, I'll let you go, and I'll see you on Friday then. Bring questions for me about evolution stuff, the chapters, all that good stuff.
Thank you.
Thank you.
Good one.
Thank you. Thank you.
Thank you.
See ya.